Question 1. Recombination. Two herpes virus strains infect the same host cell. One strain carries a gene for drug resistance and the other carries a gene for increased virulence. After co-infection, a progeny virus is isolated that possesses both traits. What viral genetic process best explains this observation? A. Antigenic drift. B. Reassortment. C. Complementation. D. Recombination. E. Phenotypic mixing. Recombination. Explanation. Recombination refers to genetic exchange between viruses with similar sequences, usually via homologous recombination. It results in a new virus containing genetic material from both parent viruses. Why not the others? A. Antigenic drift involves point mutations, not gene exchange. B. Reassortment requires segmented genomes, e.g. influenza. C. Complementation provides functional proteins but doesn't alter genome. E. Phenotypic mixing alters viral coding, not genome. Question 2. Reassortment. A new influenza A strain causes a pandemic. Genetic analysis shows it contains gene segments from avian, swine, and human flu viruses. What genetic mechanism is most likely responsible? A. Antigenic drift. B. Recombination. C. Reassortment. D. Phenotypic mixing. E. Complementation. Correct answer, C. Reassortment. Explanation. Reassortment happens when segmented viruses, like influenza, swap gene segments during co-infection. This process is responsible for antigenic shift, which can cause pandemics. Why not the others? A. Antigenic drift involves small mutations, not gene swapping. B. Recombination doesn't involve segments. D. Phenotypic mixing affects only surface proteins temporarily. E. Complementation doesn't permanently change genome. Question 3. Complementation hepatitis D virus, HDV, is unable to infect hepatocytes unless hepatitis B virus, HBV, is present. What is the role of HBV in this situation? A. It undergoes recombination with HDV-B. It enables reassortment with HDV-C. It provides structural proteins for HDV. D. It causes phenotypic mixing with HDV. E. It initiates reverse transcription of HDV genome. Correct answer, C. It provides structural proteins for HDV. Complementation. HDV is defective and requires HBsAg surface antigen from HBV to form an infectious particle. Why not the others? A. No gene recombination occurs. B. Reassortment occurs only in segmented viruses. HDV isn't one. D. Phenotypic mixing is temporary. HDV's dependency is constant. E. Reverse transcription is a feature of retroviruses, not HDV. Question 4. Phenotypic mixing. Two viruses infect a cell simultaneously. One virus has genetic material encoding for a non-infectious capsid, while the other virus has infectious capsid proteins. A progeny virus is produced with the genome of the first virus, but the capsid of the second. What process best explains this? A. Complementation. B. Phenotypic mixing. C. Reassortment. D. Recombination. E. Antigenic drift. Phenotypic mixing. Phenotypic mixing occurs when the progeny acquire the capsid of another virus while keeping their own genetic material. This can change infectivity temporarily, but it does not alter the genome. Why not the others? A. Complementation protein sharing, not structural code exchange. C. Reassortment only for segmented viruses. D. Recombination genomic, not structural. E. Drift equals small mutations. Question 5. Mixed mechanism question. Which of the following genetic mechanisms is most likely to result in a permanent change to the viral genome? A. Complementation. B. Phenotypic mixing. C. Reassortment. D. Antigenic drift. E. Both A and B. Correct answer, C. Explanation. Reassortment permanently changes the viral genome by exchanging segments between two viruses. Why not the others? A. Complementation temporary, non-genomic. B. Phenotypic mixing, surface change only. D. Antigenic drift, slow point mutations, not sudden genomic change. E, A, and B neither cause genome alteration. Let me know